Hello, my name is Mackenzie Roach, and today I'm going to be discussing the cell organelles as well as the cell cycle. I'll start off by showing this diagram of the cell as well as each cell organelle label. The mitochondria, to start, is of course the powerhouse of the cell. As we all know, it is the organelle that produces ATP. The plasma membrane that surrounds the cell acts as a barrier to select what can and can't enter in the cell. It also establishes what exits the cell as well. The plasma membrane has a phospholipid bilayer that allows that to happen. The plasma membrane also provides a stable environment for the cell as well as structural support. The next organelle that I will discuss is the ribosomes. Ribosome can be in the cytoplasm or it can be studied on the rough endoplasmic reticulum. The rough endoplasmic reticulum is a membrane that ribosomes are attached to. Cytoplasm is the free space inside the cell. While the rough ER synthesizes and modifies proteins for secretion or delivery to other organelles, the Golgi apparatus, which is right here, further modifies, sorts, packages, and transports the proteins to their final destination. The next organelle that I'll discuss is the nucleus. This is the nucleus. The nucleus encloses and houses and protects the genetic material, DNA, of the cell. Inside of the nucleus is the nucleolus, which generates rRNA, which stands for ribosomal RNA, which then turns into ribosomes. Another organelle that I'll discuss are the peroxisomes. The peroxisomes carry out different metabolic processes such as the breakdown of fatty acids and the detoxification of hydrogen peroxide. The smooth endoplasmic reticulum, which is right here, the parts of the endoplasmic reticulum that do not have ribosomes. The function of the smooth endoplasmic reticulum was to synthesize lipids, metabolize carbohydrates, detoxify the cell, and store calcium ions. Here is the microtubule. The microtubule is a protein filament. This is the centrosome. The centrosome is the organizing center for the microtubules. And the centrosome is made up of two centrioles, each positioned at a right angle. Centrioles organize microtubules during cell division. The last structure that I'll discuss is the lysosome. The lysosome is what takes away the waste material and breaks down organelles that are no longer needed. We will move on to talk about the cell cycle. This is the diagram which I've chosen. In interphase, we start out with G1. And in G1, we only have one central, as you can see here. It is where the cell begins to grow. The next part of interphase is the S phase, where DNA and centrioles duplicate. The next part of interphase is G2, where the cell prepares for division and there's two centrioles present and ready. That completes the interphase, and now we move on to mitosis. The next phase is prophase. In prophase, the chromosomes begin to condense, the spindle fiber forms, and the centrioles move to opposite sides of the cell. The next phase is metaphase. Metaphase, the chromosomes line up at the cell's equator, which is known as the metaphase plate, and the centrioles anchor the spindle fibers that attach to the kinetochore. The next phase is anaphase, where the centrioles act as anchors. The spindle fiber begins to shorten and pull the sister chromatids apart to either side of the cell. Final phase is telophase, where the centrioles remain at opposite sides of the pole as the spindle fiber breaks down and nuclear envelopes begin to form. Finally, in cytokinesis, the cell begins to divide its cytoplasm, creating two daughter cells, and the centrioles are distributed so that each cell has a pair of centrioles.